SwayamPrabha. Digital India. Educated India.
Welcome back to the fifth lecture of this week, final lecture of this week. So, when we ended the fourth lecture, I told you let's get a feeling of the timing. So, if you follow this graph here, you will observe something. So, these are the timing which are given 0.0, right, it is starting at sinoarterial node, then followed by it travels, even if you see the time 0.03, by the time it reaches here it is 0.16, and if you follow it you will say 0.21, which is the maximum, and here 0.22, which is the last point. So, if you see from here to travel here, which is of course following a convoluted route, it takes lesser time as compared to here. So, this is what gives you a feeling of time, and this impulse is getting repeated. Okay, and actually it should be split up like this. It is getting repeated, and this repetition continues. Okay, so this is how it is. So, this process of repetition. So, there are two electrical events and one mechanical event which is happening. So, this is the conduction system electrical event, and contraction system, which is the action potential of the cardiac myocytes. These are the two electrical events, followed by the real contraction by cardiac myocyte. So, you can say two electrical events EV and one mechanical event ME. This is what dictates the whole process. So, if we move on to the next slide. So, now you can visit this video, that will kind of give you an idea about some of the disease conditions and everything. Now, this is the fundamental question: what is ECG or EKG and how it is linked to the conduction and contraction system? So, we have talked about action potential of pacemaker, right? This is the action potential of pacemakers. We have talked about the action potential of cardiac myocytes, and we talked about this is where the induction is happening. Okay. So, this is getting spread into this cardiac myocytes, right? Up to this it is clear. Now, I told you, I introduced two things. I told you about conduction system electrical activity, contraction system electrical activity, and contraction system mechanical activity, and electrical activity. Okay. These electrical activities and these mechanical activities has to be integrated. Okay. And as a matter of fact, this whole structure is kind of an electrically active structure. So, there are three ways you can look at it. One of the ways is you analyze the action potential of individual pacemaker cells or the whole pacemaker circuit. You analyze the action potential of individual cardiomyocytes or the population of cardiomyocytes, or when you integrate these two electrical activities, this heart behaves as a complete electrically active system, and mixing of these two things leads to a kind of, let's put it like this. So in other words, what is happening is that, say if this is the heart, so for example, so what is happening that because of the electrical activity and movement, so this whole part initially getting depolarized, soon it is getting polarized, again now this part getting polarized, the next part is getting depolarized, then this part is getting depolarized, then followed by the next part getting depolarized, and well the previous one getting polarized. So as if it will look like there is a series of dipoles which are traveling along like this, as if there are dipoles which are moving positive, negative, positive, negative, positive, negative, then again positive, negative, positive, negative, positive, negative. So this motion of the dipoles leads to an summation of electrical activities which actually you see in the form of what we call as pacemaker, and I'll come to the pacemaker soon, but just before that let's kind of see through action potential of the cardiomyocytes. So these are the different phases. This is what I haven't shown you. So if you look at it, they are sitting at around, somewhere around, these are the 4 different phases which they are involved. The pacemaker potential, phase 1 and phase 2 do not occur. In the phase 4, which is, so phase 1 and phase 2 does occur in the action potential what we will be dealing with the cardiomyocytes. So phase 4 is the pacemaker potential, which is out here, because a slow inward current of sodium and voltage gated increase in calcium conductance via T channels, which is the phase 0 is the depolarization phase we talked about, as opposed to the ventricular muscle action potential. And if you compare this with the ventricular muscle action potential, which is slightly different. So this is your cardiac pacemaker. Sodium, this is not the fast gated voltage current, calcium with the phase 0. If you just follow previously, so this is phase 0 where the calcium is coming into play, okay. It's a little bit of a sodium followed by a calcium, okay. And then of course the flux. So there are 3 different currents what we're dealing with: potassium current, calcium current mostly, and a little bit of a sodium current which is involved in it. Now if you see the conduction, action potential conduction velocities of the cardiac conducting system, you will see these are the conduction velocities at different part of that circuit. The circuit what I have drawn. So this is how it looks like: SA node, intra-arterial pathway, AV node, bundle of His, Purkinje fiber, myocardium, and they have different velocities. This is for your academic interest, you can go through it. Next, action potential of cardiac myocyte. Now comparison, that is a contractile system. Cardiac myocyte can be divided into work cells and pacemaker cells. The work cells have the large stable resting membrane potential and display prolonged action potential with plateau phase. On the contrary, the pacemaker cells have an unstable resting membrane potential. We have already talked about it. You can read through it and that will be helpful. Now this is how the cardiac myocyte cells looks like. With all these striations, look, they look really beautiful, and these are in a dish how they look like. If you see the action potential now of cardiac myocyte, they are coming from -90. This is that whole sodium phase, okay, followed by a calcium influx here, followed by the potassium. So you can divide, and if you look at it, so this is what is happening: sodium, 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 sodium phase, sodium entry, followed by calcium entry, followed by potassium exit, and further potassium entry, which is happening here. So this is the fundamental difference between monophasic action potential of cardiac muscle cell or cardiac myocyte as compared to the pacemaker cells, okay. So this is something which I want you to kind of compare back and forth. Now further detail, if you look at it, this is what I was trying to tell you: sodium entry, calcium in, potassium out, and then here again the pumps takes the role of ensuring calcium in and sodium out and calcium in. Sorry, calcium, actually it should be out, because calcium out means it is going to the sarcoplasmic reticulum. So I told you that in sarcoplasmic reticulum, the calcium is ejected out and it is being pulled back. And these are the different phases. This you can compare, that 0, 1, 2, 3, 4 phases, you can compare it with. So this is where the comparison comes into play. The phase 1 and 2 does not occur. Okay. Now phase 1 and 2, where is that calcium in and out, this phase does not happen in the pacemaker cells. So remember this part. So this is the basic fundamental difference when we talk about, and if you look at these one, these will be following something like this. And parallelly there will be a mechanical event of contraction which will occur. Membrane current that generates a normal action potential. This is how this whole thing kind of functions. You can go through it, repolarization, and that will give you a better idea about it. And during this process, what you observe is, this is that sarcoplasmic reticulum, which is regulating this calcium exit and entry, and this I have already talked to you in the muscle cell. So this is where the calcium is being regulated. So there is a spike of calcium which ensures that the contraction happens. You remember when we talked about the muscle contraction, I told you there are sarcomere and there is a movement of that actin and myosin filament. So this is where the actin and myosin filament are happening. So this is where two electrical events, EVs, leading to one mechanical event. This is where the ball game is. And this is also where your two critical receptor comes into play, which are ryanodine and dihydropyridine DHPR receptors, and both of them are calcium activated channels, they coupled with each other, okay, DHPR and RYR, ryanodine receptor and DHPR receptors. Okay. So next, these are some of the superimposed curve of calcium. This is for your academic interest. You can have an idea how the calcium, especially in confocal microscopy, these calcium waves are being measured using different kind of dyes, and I just wanted to give you a feel that how the calcium waves are being measured, and basically this is a ratiometric measurements of calcium waves, which kind of gives you an idea that how this calcium influx moves through. And this is again a very similar examples where you see this color change. So this is where the calcium influx is kind of, calcium wave is kind of traveling through. These are the measurements which are very critical. And action potential and force comparison, if you look at it. So this is the classic action potential of cardiac myocytes, and if you look at the force generation, so this is the contraction force, and this is the time window of the contraction forces, whereas comparing it with the skeletal muscle. So if you look at it in skeletal muscle, things happen in a shot, one shot, tuck, big force, whereas here the force is distributed over a period of time. So if you look at the time window, it almost lasts for 300 milliseconds, whereas here in the skeletal muscle, it lasts much, almost just 50 milliseconds. Okay. So these are some of the fundamental differences what skeletal muscle and cardiac muscles have, skeletal versus cardiac muscle, and this is very critical for you to kind of understand these basic fundamental differences. And this is what in the beginning I was telling you, that this phase is the one which is helping it to rest, because it has to beat a gigantic amount of time all along its life, and it has to pump 8,000 liters of blood every day. This is not an easy feat to achieve. It must have taken evolution forever. So if a comparison of action potential from pacemaker and ventricular cardiomyocyte cells, these are the basic differences what you needed to work out. Cardiac action potential, large stable resting membrane potential around minus 80 millivolt, right. Smaller unstable potential around minus 50 approximately or minus 40 millivolt. It has a prolonged plateau with a prolonged action potential with a plateau. So, you remember this, something like this. Spontaneously depolarized. So, this is spontaneous, completely spontaneous and intrinsic and it controls the intrinsic electrical activity of the heart. Right and left atrium and ventricles, the locations are here found in the SA and the AV nodes. This is the location one. Ionic movement are sodium, calcium and potassium movement. In contrast to the cardiac myocytes actin potential, there is no inward current of sodium ion. This is the very fast moving, fast activating sodium current is missing. This is missing in this. Okay. This is the basic fundamental comparison side by side view. You remember I told you that this part is missing, 1 and 2 phase are missing. You do not see that. Okay. Otherwise, you see everything. You have all these phases are there, common phases 4, 0, 3, 4. This comparison is very critical for you people to have a very clear understanding of it, and that is the reason why I kind of put it here. What is ECG or EKG? How is it linked to the conducting and contraction system of the heart? Electrocardiogram, if you look at it, ECG represent a summation of action potentials across the entire heart. ECG reading includes interpretation of the following P wave, PR interval, QRS complex, QT interval and ST segment. So, it is essentially the whole heart is an electrically active unit. This electrically active unit, the electrical impulse move from SA node to AV node likewise, and in that process there is a contraction, the electrical activity is transmitted to the cardiomyocytes and that leads to an mechanical action. So, there are two electrical events superimposed over each other which is regulating the whole heart. Now, the summation of everything is your ECG or EKG traces, and if you look at the ECG and EKG traces, so this is how the ECG is a graphic recording for the changes occurring in the electrical potentials between the different sides of the skin as a result of cardiac activity. So, all these things are happening on the surface, you are making the measurements on the surface. So, now this is what I was trying to tell you. It is almost like the dipole causes current flow in the surrounding body fluid between the ends of the heart, resulting in a fluctuating, this is very important, fluctuating electric field throughout the body, and that fluctuating electric field travels like this: positive, 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 negative, 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 positive, positive, negative, positive, 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 negative, negative, negative likewise, and detecting electric field using surface electrode. Okay. This is how this whole ECG thing happens. What we essentially measure in the ECG or EKG using electrodes on the surface of the skin, we detect the voltage of the electric field. Remember the voltage of the electric field. This is what provided by the echocardiogram, and this is how this picture will help you to understand. This yellowing what you see is the area which is active, electrically active. So, the first phase of the ECG is this much. When both of them are there, you see the first wave which is the P wave, followed by these two lower ones where you see the ECG has become almost like this, followed by it spreads further out here, then you see something like this, and again the whole process repeats. So, this is how the process continues, and you can also see in different color codings. So, this is the area which is active, followed by this is the area which is active, followed by this is the area which is active, followed by this is the area which is active, followed by this whole thing gets active further, and then it narrows down and then nothing is active. So, this is the electrical activity pattern, and if you look at it, millivolts and with respect to time, and this pattern continues, and this is the QR. So, this is that P wave what we talked about in the green. This is the Q wave what you observe in orange color. This is the R wave which is this whole thing, R wave in the red. Then you have in the pink, you have the S wave which is S wave. Then you have the T wave, which is the last wave what you see. And in the normal heartbeat, this is like this. In the fast heartbeat, it's like this. In a slow heartbeat, it's like this. And irregular heartbeat, you do not see a pattern. So this is the one which worries the hell out of a doctor whenever you see a irregular heartbeat. Okay. Now P wave basically signifies the depolarization of atria, the vessel. PR interval, time from the onset of atrial depolarization P wave to the onset of ventricular depolarization, which is the QRS complex. Duration of the atrioventricular conduction is around 120 to 200 milliseconds. The QRS complex, the duration of depolarization of ventricle is less than 120 milliseconds. The QT interval, the depolarization and repolarization of the ventricle is less than 420 milliseconds, potentially to 440 milliseconds in the case of women. Okay, have a look at this one. And then the PR interval, duration of the ventricular cardiac cycle and indication of the ventricular rate. So this is how you can correlate the two different action potential of ventricular and atrial cardiomyocytes and the ECG trace. Okay. What ECG tells? This is very important. Anatomical orientation of the heart, ECG can tell you. Relative size of the heart chamber, it can tell you. It can tell you the heart rate, it can tell you about the rhythm, it can tell you the origin of excitation, it can talk about the spread of the impulse, it can talk about the decay of the excitation, it can talk about the disturbances of the above event, not the cause, not the cause. This is important. Electrocardiogram, ECG gives no direct information about the contraction and the pumping efficiency of the heart. In terms of the pathology, you could have blockage on these pathways at different spots, as you could see shown in red, and those blockages could lead to different kind of problems. The temporal correlation, if you look at it in terms of time, you can really go through it. That will give you an idea that one action potential is lesser than the whole interval of the P, Q, RS and T complex. Okay? And this is how it looks like when you are measuring the ECG thing. And there are certain control units which I am not going to deal today on this, because there are obvious reasons, because I still haven't talked to you about sympathetic and parasympathetic in the system. Once I talk about the sympathetic and parasympathetic, I will talk about it. And then talking about the artificial heart, what we talk about, these are those artificial heart in case you have to replace the whole heart. These are some of those models which are currently being tried out by Apcor Hearts and other companies, where you can replace the whole pumping machine with an artificial heart. So keeping all this in mind, I am closing on this week. This is what all we have to cover about the cardiac physiology. So I hope this will help you, these 5 lectures, to get an overall idea about the cardiac system and how the heart works. Thank you.